Okay, guys, part two of our photosynthesis screencast. This is where we left off on part one, which was just talking about the different reactions of photosynthesis. We are going to start with the light reactions. And for us to start with the light reactions, we need to think about, first of all, the overall reaction of photosynthesis, which is the opposite of cellular respiration. Carbon dioxide and water in with sunlight, of course, as the energy gives you C6H12O6, which is your glucose, and oxygen, which is your waste product. For the first thing we need to think about how plants capture the energy of the sunlight, because our first set of reactions are light dependent, meaning they're the ones that use the sun. So we're looking at CO2, light, and chlorophyll, which is the pigment in plants that make them green. Basically, when we see green, what we're seeing are those chlorophyll pigments. Okay, now plants gather the energy with these pigments. These pigments are light absorbing molecules. We have two main types, chlorophyll A, chlorophyll B. We don't need to go too nuts with that. But what we do need to look at are the kinds of light, or I should say the colors of light, that are absorbed by plants. And if you look here, this gives you the differences between chlorophyll B and chlorophyll A. But what's really important, and here's our color spectrum: violet, blue, green, yellow, orange, red. What you'll see from this graph, right? Estimated percentage of absorption. Here in the green range, we go down to almost zero. So chlorophyll A and B together are going to absorb violet, blue, and then kind of coming off here, yellow, orange, and red wavelengths. That is why we see green, because the green light actually is reflected off of the leaves and what goes into our eyes. Now, when light is absorbed, right, that is energy. So when chlorophyll absorbs that light, that energy is transferred to electrons. Just like in the electron transport chain of cellular respiration, right, those electrons get excited. They're taking the energy that is extracted out of the glucose and moving it to make ATP. We're going to see a similar thing happen here, where the electrons in the chloroplasts actually take on the energy of that light, become high energy electrons, and then provide the energy for the plant to create glucose. Okay, so let's think about chloroplasts for a moment. Do you guys remember we actually saw the chloroplasts in the Elodia plants in one of our microscope labs? That's what we are really thinking about. In these chloroplasts is where this entire process is going to happen. This is a nice model of a chloroplast. Within the chloroplast, we have these stacks, right? So if you guys remember the video from last week, the teeny tiny pigment pancakes, that's what these are, teeny tiny pigment pancakes. These are called thylakoids, sac-like photosynthetic membranes. And it's in these membranes where a lot of the magic is going to happen. So we have single thylakoids, we have stacks of thylakoids, and then we have stacks upon stacks of thylakoids. So a stack of thylakoids is a granum, and then all of those stacks within the chloroplast are grana. I can rarely remember which is which, grana, granum, it's fine as long as you know that. And then, like I said, it's in the thylakoid membranes where a lot of this is going to occur. Proteins in the membrane called photosystems, just like the electron transport chain of cellular respiration, that is where this process is going to happen. So here's a nice model of a chloroplast showing you what goes in and what comes out. So here we have the light-dependent reactions, which happen in the thylakoid, specifically in the thylakoid membrane. Water and light go in, oxygen comes out as waste, ATP and NADPH are produced. So a little bit of energy and electron carriers that are going to get sent over here to the Calvin cycle, which will use the energy, the electrons, and carbon dioxide to give you your sugars or your glucose. Now, electron carriers, just like cellular respiration, right? So if you understood the electron transport chain of cellular respiration, it's almost the same exact thing, except it's in a different location, and the proteins, right, those transport proteins or carrier proteins have different names, but essentially it's going to look just like the electron transport chain of cellular respiration. So NAD plus, sorry, NADP plus. I always think P for photosynthesis, right? So in cellular respiration, it's NAD plus. Um, in photosynthesis, NADP plus. But you see you have that plus. So this is ready to take on negative charges. So it takes two high energy electrons right along with their hydrogen and gives you NADPH. Okay, so here we are, light-dependent reactions. Looks very, very similar to cellular respiration. And you can see, right, here we have our ATP synthase in the thylakoid membrane, which again, phospholipid-based. Here are your photosystems, which are your carrier proteins. Photosystem 2 actually comes first. It was discovered after photosystem 1. Doesn't really matter. Electrons come in, right? So there are electrons here. Those electrons take the energy from this light. They get excited. As they get excited, they go through this electron transport chain. And remember, this is also where you use your water. That water is what basically provides the electrons as you're going through this process. So the water gets broken into electrons, which go through here, hydrogen or H plus, right? Hydrogen ions, and then oxygen. Remember, oxygen is the waste product of the light dependent reaction. So that's everything right here. Every time a water molecule is used, it sends its electrons through here. So as the electrons move, they're constantly replaced by the electrons from the water that the plant is using. Oxygen is released into the air, that's the waste. Hydrogen builds up, comes across the membrane. Now this is talking about stroma, right? So if you think about the chloroplast, the liquid inside the chloroplast is called the stroma. So these hydrogen ions are moving from the stroma into the inner thylakoid space. So it means they're crossing the thylakoid membrane. And we're going through, right? And as they move through, they get re-energized. NADPH picks up these electrons, which get re-energized by light along the way, with some of the H plus, gives you NADPH. And then very similar to cell respiration, all these hydrogen ions build up here and have to get out. The only way they can get out is through ATP synthase, and that's what gives you your ATP. I wish I could ask you guys if this makes sense. Think about this as very similar to the electron transport chain of cellular respiration. It's essentially doing the same thing, except water is your reactant, and it's water that's providing all of these electrons that are constantly being re-energized by the light as it's being absorbed by the chlorophyll. And that's basically the light-dependent reactions. All of the products of the light-dependent reactions go into the Calvin cycle, the ATP and the NADPH. So energy and energized electrons, which are going to help the plant make sugar. These reactions are called light independent. Back in the day, they used to be called the dark reactions, but they can happen at any time. They can happen in the day or at night. However, they do not require sunlight. Now, here's the equation for the Calvin cycle. Again, this is endergonic because it is using energy to make sugar. Endergonic, light independent reactions use ATP and NADPH made during the light reactions to provide energy for the synthesis of glucose and other organic molecules from inorganic carbon dioxide and water, right? So here's your NADPH, here's your ATP plus CO2, that's the other part of the overall photosynthesis reaction, gives you a molecule of glucose, NAD plus, which is going to go back to pick up more electrons, ADP plus P, which is your used up ATP, and water, which will end up going back into the plant just to be used for other processes. Now, this is a little ugly right here. And just like, the Krebs cycle, there's a ton of rearrangements of carbon compounds that are happening here in order to give you a molecule of glucose. There are a few things that I'm going to ask you to know, but mostly we're just going to kind of skim the surface of these chemical reactions. The first thing that happens is CO2 fixation. Fixation is a fancy way to say it goes from a gas to a solid. So CO2 diffuses into the stroma, right, with the help of an enzyme, combines with molecules of something called RUBP, ribulose biphosphate. That will give you 6 carbon molecules. They will be unstable, split into 3 carbon compounds, and then those are converted into G3P, right? Does that sound familiar? Should, from cellular respiration. Step 3, 1G3P leaves the Calvin cycle and is used to make organic compounds. So that's where your glucose comes in, right? Because you're making 1 molecule of glucose with this process. The rest of them are converted back into RUBP and go back, to the beginning to fuse with carbon dioxide to get you G3P to make glucose. So it's kind of an inefficient process. For every 6 molecules of CO2 that go into the Calvin cycle, you get 2 G3Ps, and then most of the G3P goes back to regenerate the RUBP to keep the Calvin cycle going. So it takes 6 turns of the Calvin cycle to produce 1 molecule of glucose. Now, a couple other things that we can chat about, and I know that maybe that seemed confusing. I did link a couple of good videos for you guys to watch, one Khan Academy and one TED Ed on the Calvin cycle. But again, as long as you know, right, simple steps, fixation, creation of G3P, 1 G3P makes glucose, the other go back into the cycle. We can keep it super simple. Okay, there are some alternatives to regular photosynthesis because plants, right, live all over the world. So they've had to adapt to different climatic conditions, specifically very dry conditions, because the structures on the undersides of the leaves that allow for gas exchange are also going to let water out. So plants want to be super careful about not losing too much water. So we have 2 alternate pathways. One is called the C4 pathway. So instead of making G3P, right, which is a 3 carbon compound, this will end up making a 4 carbon compound, which will then end up going into the Calvin cycle. And the CAM pathway, crassulation acid metabolism. These are true desert plants. They'll only open their stomata at night, so they'll only do gas exchange at night in order to prevent water loss in hot conditions, dry conditions. Okay, and this is just a little bit about what is going to impact photosynthesis. Obviously, you need light. At some point, you'll reach the maximum rate of photosynthesis with light. Temperature, increasing temperature will accelerate the rate of photosynthesis until you reach the point where enzymes can no longer function, and then you see a big drop. Carbon dioxide as well. Increasing carbon dioxide will increase the rate of photosynthesis, but again, you'll reach a maximum. So if you think about greenhouses and places where people have total control over what plants are receiving, carbon dioxide levels get increased at night in order to increase photosynthesis, the light independent reactions, and especially at night because you don't want to increase CO2 when people are there because that could be detrimental to humans. Okay, and now we're getting into some of these rearrangements. We don't need to go there. We can actually wrap this up right here. So I'm going to post this. If you guys have questions, of course, I'll be available to answer them. I just went through, several days of stuff in one super fast lecture. So let's chat about it. Give me feedback, and I'll talk to you guys soon.